Are these coffee beans any different from ordinary coffee beans? Well, to answer that very question, I've come here to the University of Guelph, to the Food Science Laboratories. So, Massimo, you were the man who was charged with finding out if the beans had changed or not. How did you start your examination?
Well, the first thing we did is we did a visual inspection of the bean, and we had two beans. We had our control, the bean that had not gone through the cat's intestinal tract, and we had the other bean, which actually was the Coffie luwak that actually had gone through the whole GI tract of the cat.
And what did you find?
Well, you can see the beans right here. We have the control bean here, and we have the Coffie luwak here.
Okay.
And first thing that we notice is that there in fact is a difference in color. We want to make sure that in fact what we are actually observing with our eye is actually what we're seeing. So we use this particular instrument here, which is a colorimeter that actually helps us to put numbers to the colors.
Okay, and what's it looking for?
Okay, it's looking for the lightness and the darkness and different hues. For example, yellow to red and blue to green.
Okay, so can we have a go?
Sure. I'll let you do it.
Okay.
So just grab the gun, and you just put the gun perpendicular to the beans.
And then just hit the button, yeah?
Exactly.
Great. So idiot-proof, really, isn't it?
Exactly.
So what have we got here?
We can see here is basically this one is higher in the yellow and the red, which gives us that brownish color or beige color that we're actually seeing for the bean. Okay,
so we think the color's different. What else did you look for?
Well, we actually wanted to determine if in fact the enzymes and the acids of the stomach were actually affecting the bean itself. And so we looked at the beans using the scanning electron microscope.
And this is the outside of the bean.
Exactly.
And I can see that one's a lot rougher. Why is that?
As it's going through the digestive tract, it's exfoliating the different layers, and we're actually seeing a smoother surface to the Coffie luwak. This is at 100-time magnification, and we can actually see that the control...
Look at it, yeah.
It's much rougher than the thing. What I find more interesting, if we go to 1,000 times, what we can see now is that we actually have pitting, which is actually occurring due to both the enzymes and the gastric acids that are in the digestive tract. We don't see that in the control. So our next step was to actually look at the electrophoretic pattern, which is looking at the protein profile.
So which one is the Coffie luwak?
Okay, the Coffie luwak is this one right here, and our control bean is this one right here.
So the bands here are a lot lighter.
Yes, the lightness, what that is caused from is that the enzymes are actually getting into the bean and they're breaking down the proteins. We also see some what we call proteolysis, or the breakdown of the protein. The proteins are being broken down to what we call peptides, small proteins, and amino acids, which are the building blocks of the protein, and so they're actually migrating up.
And how will that affect the flavor?
Well, there's been some work that's indicated that the level of amino acids has a direct bearing on the flavor, because during the roasting they react and they cause certain flavor compounds. So we actually have more of them in the Coffie luwak, which will probably lead to a difference in flavor.
Better flavor?
Well, that we don't know as yet.
Can you use this then as a kind of genetic fingerprint for the Coffie luwak bean?
Exactly, because proteins are actually the gene expression, what we can do then is actually look at these two profiles, and in cases where there might be question as to the authenticity of the actual coffee, is it Coffie luwak or not, we can actually run a test like this and actually say, in fact, it is from Coffie luwak or it's not, or has there been adulteration? Do we see some of the control bean, native bean, in the Coffie luwak? So we can actually use it for regulatory purposes.
So this is like the forensic science then of the coffee bean?
Exactly.
Now, Massimo, many people are going to be concerned that these coffee beans have been collected from the feces of an animal. Did you find any nasty bacteria on them?
In fact, I thought the same thing. When the beans first came in, what I did is I put on gloves, and because I didn't know what was actually on them, so they were quite correct in being concerned at that point.
What did you find, though?
So what we have here is the total bacteria that are actually on the bean themselves. Then we have the total plate count, then we go to what are called the enterics. The enterics are more the bacteria of concern. They're more the pathogens. Those are the ones that we should be more concerned about. So we take a subculture of these, and what we're actually looking at is This is the control, and this is the Coffie Luwak here.
Right.
And what actually we find is that the control is actually more contaminated than the Coffie Luwak.
There's loads here. I can't see any on this one, actually.
There's just a few little colonies on there. So what we did then from that point was to take it one step further and look at a particular type of bacteria which are considered the enterics, which are the coliforms. And you can see the actual coliforms here.
Okay.
This coliform is the control.
Wow.
And you can see how many there are on there. And you can see that we only have a few on the Coffie Luwak.
Yeah, so that's a big difference. So what's happening then? How come they're being killed when they pass through the animal?
Well, what is happening, I think that we know from the actual processing of the bean that the people take it and they do a washing procedure. Now, this washing procedure of the bean can actually wash away some of the pathogens, much like when people wash the vegetables. So it's not
when it's passing through the animal that they're being killed off, it's afterwards that when the people are collecting them, they're being washed off.
Exactly.
So we've talked about sort of the structure and the sort of microbiology, but what about the actual flavour of the coffee? Was there any difference in that?
Well, in fact, what we did was we took a look at the volatiles that are in the coffee themselves.
So the volatiles are the substances that give it the flavour, are they?
Absolutely. Well, we've actually seen that the profiles are very different.
I can see that, it's quite clear. So what does that mean, though? What does that tell us?
Well, it does tell us that the taste, in all probability, is going to be different between them. So going through the intestinal tract of the cat has produced a difference in probably flavor, which can be translated into flavor.
But now you know the science behind it. As a consumer, would you pay $600 for a pound?
Well, if I was Donald Trump, I might.
And in case you're wondering, Jay and I did get to try some of the Coffie Luwak coffee, and it tasted pretty damn good.
Well, and the fact is that when we drank it, it had been sitting around for a little while, so it wasn't super hot as coffee should be. And nonetheless, it was dramatically different from the control coffees we had. So I'd love to drink it hot sometime.
But have you bought it since?
No, I bought it. It's $600 a pound.